Right, well this is our second video on the kidney, so we're carrying on from where we left off in the last video. We are hopefully going to get through osmoregulation in this video, leaving us with just the question pack to finish off in the last video. Okay, so we're going to start with a recap of what we did last lesson. So what I'd like you to do is to quickly sketch down this diagram here with all the parts of the nephron. And then I want you to write next to each part what happens in that region of the nephron up to the loop of Henle where we're up to. Okay. Okay, let's review what you should have next to each part. So at your glomerulus and Bowman's capsule at the start of the nephron here, we should have some information first about the glomerulus, which is this capillary network, noting that the arteriole supplying the network has a wider lumen than the arteriole taking the blood away from the capillary network, which has a narrower lumen. That creates high hydrostatic pressure in that capillary network, so the blood is under high hydrostatic pressure. The walls of the capillaries, of course, are permeable. We know that about capillary walls anyway. But to add to that, the Bowman's capsule is described as the molecular sieve because there are pores in the basement membrane between the capillaries and the Bowman's capsule wall. And the Bowman's capsule wall is made out of cells which are called podocytes, and there are gaps between the feet of the podocytes. So we've got pores then between the blood and the space that the filtrate moves into. So this is the site of ultrafiltration, which is the process by which all small molecules in the blood plasma are forced out of the blood, forming the filtrate in the nephron. Molecules such as glucose, ions, amino acids will be forced out, and of course urea, which is the waste product we want to get rid of in the urine. So this is the process of starting to form the urine. The solution now in the nephron we refer to as the filtrate, because lots and lots of things change about its constituents in terms of how much water, how much ions, how much of each of these molecules are found in it before it exits the nephron via the collecting duct at the end of the process as urine. Okay, so that's the first section of the kind of things you should have there. Moving into the second part that we looked at, this was our proximal convoluted tubule. The walls of the proximal convoluted tubule, I'm hoping you've written, have got microvilli to increase the surface area of them for absorption. They've got many mitochondria to provide ATP for absorption of all the things that we want to take back from the filtrate into the blood, things like glucose and your amino acids, and also are closely associated with many capillaries to give it a large surface area for that exchange. So the proximal convoluted tubule is set up to reabsorb most of the important molecules that you would not want to lose in the urine, and they are absorbed back into the blood here. Okay, but there's still a lot of water and ions to be reabsorbed back. Okay, and that's what the next parts of the nephron are all about. So we worked our way to the loop of Henle. Now the loop of Henle and the countercurrent is probably the more difficult bit. So next to the loop of Henle, I'd be really satisfied if you'd remembered this idea of the countercurrent of the filtrate down the descending limb, and after a quick turn, like a hairpin turn, then the flow of the filtrate is in the opposite direction as it goes back up the ascending limb. And that process maintains a water potential gradient for the movement of water out of the descending limb all the way down the descending limb, remembering that that's the only limb that is permeable to water. Okay, and we explain how that was produced through the movement of sodium and chloride ions from the filtrate. As you turn the hairpin first by diffusion, so down a really strong concentration gradient by diffusion, and then as you move further up the ascending limb, starting to be actively transported out. This will mean that wherever in the medulla you are, the water potential in the medulla will be lower than the filtrate just next to it in the descending limb of the loop of Henle. So that means water is always drawn out by osmosis down the whole of that limb. Okay, so that's where we got up to. So we're now moving on to the distal convoluted tubule and the collecting duct. So all of this that we've looked at so far works in exactly the same way all the time, but what happens in the distal convoluted tubule and the collecting duct is something the body can vary. So here, both of these regions, there is water reabsorbed from the filtrate back into the blood, but how much is reabsorbed can be modified depending on the body's needs. So if there is lots of water in the body and we've got an excess of water, then we don't need to reabsorb the water from the filtrate in the distal convoluted tubule and the collecting duct, and we can leave it and it will then go and produce a very large dilute urine. But if we are a bit dehydrated, then we can make changes, or the body can make changes to the walls of the distal convoluted tubule and the collecting duct to ensure that we reabsorb a maximum amount of water back into the blood, so we don't lose very much in the urine and we produce a really concentrated low volume urine. So that's what today's video is all about. Okay, so if you have a look at the distal convoluted tubule in your notes, you'll see there's not a great deal that we need to know about it. So in your keynotes, we are now here at the top of page 7. Okay, so at the top of page 7, it talks about what we need to know about what happens in the distal convoluted tubule. Well, the distal convoluted tubule structure is just like the proximal convoluted tubule. So it has the same adaptions, many microvilli and many mitochondria, okay, because these cells are also adaptive for absorption and they do actively transport a lot of sodium and potassium to facilitate the movement of extra water for reabsorption or not. So the permeability of the cell membranes is affected by the hormones that we're going to talk about, allowing some osmoregulation. So how much water is reabsorbed by these cells, it can be varied by the body in the process of osmoregulation, regulating the amount of water in the body. So all you need to know really is the idea that they've got many mitochondria, and many microvilli and many mitochondria, and why, and be able to link that to active transport of ions. Remember that if you move ions into the cell, that will lower the water potential and water will move in. So the more application points. Okay, so the convoluted tubule just adjusts the filtrate concentration by actively transporting sodium and potassium into or out of the tubule. So if they move the ions into the tubule, then you get a lower water potential in the tubule, so water would stay, not be reabsorbed. If they move ions into the cells, then that would draw water out of the tubules. So no direct questions about this section, it would just be application of knowledge. Well, there are a lot of questions that are in the collecting duct. I'll just show you, I've bobbed a couple of slides showing the distal convoluted tubule in your PowerPoint, just saying that really all you need to know is it's the exchange of ions, mainly by active transport here, that regulate the amount of water being either reabsorbed into the blood or not reabsorbed into the blood. Okay, so we're moving into the collecting ducts. Now there's a lot more exam-based questions on the collecting ducts. Here is where the main osmoregulation takes place. So the effect of osmoregulation takes place. So you can see that if we cut through the medulla, there are absolutely loads of collecting ducts. Okay, so they collect together from many different nephrons, and then of course at the end of the collecting ducts, that's where the urine leaves the kidney. Now, the collecting duct walls can be not permeable or permeable. Okay, now remember the medulla tissue is salty, and the medulla tissue is always salty because of the sodium and chloride ions that leave the loop of Henle. So that's what creates that condition of a salty medulla tissue. Yeah, so it's got a very low water potential compared to the inside of the filtrate. So there is always a water potential gradient for water to be reabsorbed from the collecting duct filtrate into the medulla, and then obviously still down a water potential gradient back into the blood. So that's normal. It's always there. But the collecting duct walls do not have to be permeable to water. So the body can change the permeability of those walls so that water can move or can't move down that water potential gradient. So it's possible for the body to make the collecting duct less permeable to water, as represented here by having a solid line. So although there is a water potential gradient still for water to move from the filtrate to the salty medulla, the walls are not permeable or not very permeable, and so less or no water can move across the walls by osmosis and be reabsorbed into the blood. But then the body can regulate the permeability of the collecting duct wall and make it really permeable to water, and in that case lots of water will move by osmosis down that water potential gradient into the salty medulla tissue, and then by again a water potential gradient back into the blood. So I want to look now at how this happens. So in your keynotes there's a little bit of that here in summary. So there is a water potential gradient for the movement of water out of the collecting duct. Okay, sometimes they ask you why. So here you might want to make a little note to yourself that that is due to the ions, the sodium and chloride ions that are moving out of the loop of Henle into the medulla tissue. Okay, so that's what creates that water potential gradient. The medulla's got a lower water potential than the filtrate in the collecting duct. So there's always a water potential gradient there due to the movement of ions out of the loop of Henle. However, the permeability of the walls of the collecting duct is also affected by hormones. So if the walls are more water permeable, water will leave the duct into the medulla and the urine will be more concentrated and have a lower volume. If the walls are made less water permeable, then less water will leave the duct into the medulla and the urine will be less concentrated, more dilute if you like, with a higher volume. So what causes a change in permeability in the collecting duct wall? Okay, well the collecting duct walls have got receptors for ADH, and I think you'll remember antidiuretic hormone from your GCSE studies. Okay, so we're going to have a little look at how this osmoregulation happens now. So we will come back to your keynotes in a minute, and you are focusing on this little bit here. Let me tell you the story first of all. Okay, So how does the body control how much water to reabsorb? And this we're concentrating on in the collecting duct, but the same thing happens in the distal convoluted tubule too. This hormone has an effect on making the walls more or less permeable of the collecting duct and the distal convoluted tubule too. Okay, so there are receptors that monitor the water potential of the blood. Okay, now this is where the exam board can link your knowledge of receptors and action potentials to this process. So we'll see a question like this in the question pack later on. Okay, so it isn't a named receptor on the spec, but at AO2 you can suggest how these receptors respond to changes in water potential in the blood. So we'll show you that question in the next video. The pituitary gland can then adjust the amount of antidiuretic hormone released into the blood. Okay. So if we have a look back into your keynotes, you can see we have a name for those receptors and a place where they're found. Okay, so the osmoreceptors, because they're monitoring water potential in the blood, and they're found in the hypothalamus of the brain. Okay, so if we go back to that picture, you can see there are two parts of the brain that you need to know about for this section of the course. The hypothalamus, okay, where the osmoreceptors are that are monitoring the water potential of the blood, and the pituitary gland, because that is the gland that secretes the ADH, the antidiuretic hormone. Okay, so let's imagine the scenario that we've got in our keynotes where there is a change in water potential and the change is a decrease in water potential. Okay, so the osmoreceptors detect a decrease in water potential. That causes the release of antidiuretic hormone from the posterior pituitary gland. Now, you can get away with just calling it a pituitary gland, but because there are two parts to it, the more accurate is to say posterior. The ADH travels to the kidney in the blood and it binds to receptors on the surface of the collecting duct and the distal convoluted tubule, and it causes them to become more permeable to water. How on earth does that happen? Well, inside the cell, it causes a series of enzyme catalyzed reactions, resulting in vesicles containing aquaporins fusing with the membrane. This, of course, would increase the permeability of the membrane of the walls of the collecting duct and the distal convoluted tubule. That means more water moves from the filtrate in the duct, in the convoluted tubule, down a water potential gradient, out of the filtrate and back into the blood. It's really important you say out of the filtrate, back into the blood. The exam board are really keen now on where things are moving to and from. And the outcome then is that more water is reabsorbed from the filtrate back into the blood. You get a lower volume of urine, which is more concentrated. So I have included this picture in your keynote, which shows that in steps. So ADH is released into the blood, binds to receptors on the surface of the collecting duct wall or the convoluted tubule wall. Inside the cells, that causes a series of enzyme catalyzed reactions that result in aquaporins, vesicles containing aquaporins, to fuse with the cell membrane. With more aquaporins in the cell membrane, the wall becomes more permeable to water. So remember, there was a water potential gradient across these walls already, and because the medulla is salty, because of the ions that have been diffused out and actively transported out of the ascending limb of the loop of Henle. Okay, so that's a nice summary diagram of how that happens. They're much more interested in this on the new spec with the aquaporins. This is the new detail that's come in. Okay. So just pause for a second. Can you remember which bits of which in our diagram on the board? So I'm hoping that you've just got a label brain for the whole thing. But then for these two, importantly, we've got B being the pituitary gland and C being the hypothalamus. Hypothalamus, where the osmoreceptors are found. The pituitary gland, of course, the tissue that secretes the ADH. Okay, so this is, of course, an example of a negative feedback loop, or two negative feedback loops. So you could arrange it in the same way as we did with our control of blood glucose, with a diagram with the set point along the centre, and then these above and below. But I put them in this format this time. Okay, so we've done this one. So low blood water levels, so that would mean that we've got a low water potential. Pituitary gland releases ADH, makes the membrane more permeable. Okay, so that's the membrane, the collecting duct of the distal convoluted tubule. More water is reabsorbed, and you get less urine and more concentrated urine. Of course, that straight away will cause the water levels of the blood to rise, so you'll get a higher water potential of the blood. And that will mean that we move to the pituitary gland stopping the release of ADH. You'll get a reduction in permeability of the membrane, less water reabsorbed, and more urine, which is less concentrated. That, of course, will then mean that the blood water levels go lower again. So, and it continues and continues in that cycle, maintaining the correct water potential in the blood. So these are examples again of negative feedback, because as we get a change away from set point or norm, it's been detected, brings about a change that brings an effect that brings you close to or back to normal, and then if you go the other way, the same thing happens to return. So you're constantly staying around a normal water potential of the blood, like that. Okay, so that's similar to the diagram we did for blood glucose control. So we've got an increase in water potential of the blood detected by the receptors in the hypothalamus, less ADH from the posterior pituitary, because the water potential is increased with loads more water in the blood than we need. Collecting duct wall becomes less permeable. You'd put in a little bit more detail about there being less aquaporins. Less water is reabsorbed into the blood, and that causes a decrease in water potential of the blood, bringing you back to normal. So this one negative feedback loop. And then at the bottom, we've got a decrease in water potential in the blood, detected again by osmoreceptors in the hypothalamus, more ADH released from the posterior pituitary gland. That would mean more aquaporins in the membrane. So the collecting duct walls become more permeable, and more water is reabsorbed into the blood and less urine produced. That would increase the water potential of the blood back to normal again. So remember, more water in the blood, the higher the water potential. Okay, so explain what happens when you drink large amounts of water. So I want you to have a think now. Stop the video and try to describe what you think would happen to bring your water potential of your blood back to normal. Okay, so you should be joining me back now. So if we say the first thing would happen is the concentration of the blood would decrease, so we've got more water. So what would happen to the water potential? The concentration of the blood is decreasing, the water potential is going up. That's detected by those osmoreceptors in the hypothalamus. The pituitary gland would stop the production or stop the release of ADH. You get a change in permeability of the tubule membranes, so you get a decrease in the number of aquaporins in the cell membrane. This would happen to both the collecting duct and the distal convoluted tubule. And the kidneys would reabsorb little water or less water. Most water then would be left in the filtrate, resulting in a dilute urine of very high volume. Okay. So here's another example of a question. Explain how the body maintains a constant blood water potential when you exercise. So this is the other way now. You're becoming dehydrated because you're sweating, losing water across the surface of the skin. So now the water potential of your blood would be decreasing as there be less water present. So here's the student response, and what I want you to do is what went well and even better if. So I'll read it through, we can mark it as well as we go. Imagine this was for about 4 marks. During exercise, the body loses water and ions in the sweat to cool the body down. The amount of water in the blood will decrease, and this is detected by a receptor in the hypothalamus of the brain. The response is more ADH released from the pituitary gland into the blood. This hormone travels in the blood to the kidney, where it binds to receptor and makes the walls of the nephron more permeable. More water reabsorbed, resulting in a more concentrated urine produced. So what do you think, first of all? You've got most of the right idea, as ever. The details not quite there. So how many marks would you give it out of 4 right now? I'm thinking about 2 or maybe 1. Let's have a look. So there would be a mark point, a linking mark point sometimes. So with this question, explain how the body maintains a constant blood water potential when you exercise. There would be a linking mark point for what would be changing and what would be changing. So your first mark point would be during exercise, in sweat, we lose water. This will cause, but you should really write about water potential. You're an A-level biologist, so the amount of water in the blood decreases. What does that mean for water potential? So you would say the water potential of the blood decreases. That's what's going to be on the mark scheme. And this is detected by, we should have osmoreceptors here in the hypothalamus, and that will be fine then. The response is more ADH released from the pituitary gland into the blood. That would be okay. You don't have to have posterior in front, so it would be okay. ADH is a recognised abbreviation, so although it's good practice to put antidiuretic hormone the first time and then use the abbreviation. The fact that it travels in the blood to the kidney might be locked in there as well. So that was okay as a mark point. There it binds to. Now when it comes to receptors on the new spec, they really want you to talk about them being complementary or specific with protein receptors to ADH. And then because it's new spec and they're so keen on the aquaporins, without how it makes the walls more permeable, we're not going to get any marks. And just the whole nephron is not affected. So you have to say the collecting duct walls and the distal convoluted tubule walls. So to improve that, you would say here, ADH binds to specific protein receptors in the cell membranes of the collecting duct and the distal convoluted tubule. This causes a series of enzyme controlled reactions which result in vesicles containing aquaporins fusing with the cell membrane, they're making the walls more permeable. Okay. More water is reabsorbed. Wouldn't be a mark point. It's true, but remember the from and to. So more water is reabsorbed from the filtrate to the blood, and then you can say giving a more concentrated urine. Sometimes you have to talk about it being more concentrated, sometimes the volume. So I would always put both, so a concentrated urine of lower volume. There's a lot of detail missing from that, so that's why it was 1 or 2 marks before. It's only likely to be a 4 marker because it's a paper 2 concept, and it's still up for grabs. Okay, so that's our osmoregulation. There's loads of AO2 on this, absolutely loads. So if you think about just caffeine, and you know that that's a diuretic, and anything you're told is a diuretic, it's going to have the opposite effect, if you like, to the antidiuretic hormone. Suggest its effect on the body. So we know caffeine, a little bit about caffeine. So what do we think would happen if someone drinks caffeinated drinks? What's going to be the effect on the nephron? Okay, with a little bit of thinking, I'm hoping you're pondering that it would have the opposite effect. So reduce the permeability of the collecting duct walls and the distal convoluted tubule walls. So that would be less water reabsorbed and more water lost in the urine. So how would a coffee drinker's urine differ to someone who drinks water? We'd expect, because they're consuming, I'm assuming the coffee is caffeine containing, they would be producing a more dilute urine in a larger quantity. Okay. Another question. What causes the volume of water in the filtrate to change as it passes along the proximal convoluted tubule? The difference in urea concentration in the filtrate before the proximal convoluted tubule and in the collecting duct, and a person to have glucose in their urine. Can you just have a little ponder about those three? What causes these? Okay, you should have finished your ponder. When it comes to AO2 on the kidney, the one thing that you've got to remember is that the examiner wants a bit of thinking, okay? Particularly about concentrations of things. So, if you fully understand what's happening in each section of the nephron, you should be able to tell them why concentrations and volumes of different molecules and total filtrates are changing. So the proximal convoluted tubule, we said, was the site of most of the reabsorption of glucose and amino acids on all the useful things we didn't want to lose from the blood. So can we explain why the volume of water in the filtrate changes as it passes along the proximal convoluted tubule? So what I'm hoping you've said is that as the glucose and the amino acids and other substances are reabsorbed by the proximal convoluted tubule, the water potential of those cells would become lower, and then as those molecules move across those cells and into the blood, that would also lower the water potential in the blood, creating a water potential gradient for the reabsorption of water. So a large volume of water would be reabsorbed, if you like, following the solutes. Yeah, as the solutes lower the water potential, as they move, water will follow by osmosis. For the second one, this is a really good question, and a lot of the data questions are based on this, as you'll see. The difference in urea concentration in the filtrate before the proximal convoluted tubule and in the collecting duct. The one thing we do not reabsorb from the filtrate is urea. The whole point of urine is to get rid of that waste product. Okay, well, not the whole point, but one of the main points. Okay, obviously urine is about balancing how much water and ions are in the body as well, but this is a waste product that's poisonous if it builds up in the body, and we need to get rid of it. So if the urea is not being reabsorbed at all from the filtrate into the blood, why would its concentration be changing? So remember, concentration is affected by two things: the amount of that molecule, but also the amount of water. So if the amount of water is changing, although the amount of urea is staying the same, the concentration will be changing. So before the proximal convoluted tubule, we will have lots of water still in the blood, still in the filtrate, having not yet been returned to the blood, reabsorbed. And in the collecting duct, at the end of this process, all that water, or most water, will be reabsorbed. So you would expect the urea concentration to be much higher at the end of the nephron than it is at the start, not because the amount of urea has changed, but because the amount of water in the filtrate has decreased as the water was reabsorbed back into the blood in different sections of the nephron. Okay, so a person to have glucose in their urine. Well, if you find glucose in someone's urine, that is not normal really from the majority, unless you're eating an excessive amount of glucose in one meal, because we know that in the proximal convoluted tubule, glucose would move by diffusion, and then we actively transport glucose back into the blood from the filtrate. So it would be if somebody has too much blood in the too much glucose in their blood, so they can't have it. It's basically not, you can't reabsorb it all by the time you finish the proximal convoluted tubule. So people with diabetes who can't produce insulin or are not sensitive to insulin, that will not uptake glucose because the insulin's either not binding to the receptors, or when it binds to the receptors, it's not having the impact of uploading glucose from the blood. They would have too much glucose left in their blood. It would not be all of it reabsorbed by the proximal convoluted tubule, and that would be why we could detect glucose in their urine. But we tend to do a fasting test when we're trying to decide whether somebody has diabetes. That means you make someone fast, and then you test their urine for blood. Sorry, not for blood, for glucose. And because after a big meal, if you've got loads of glucose in your blood all at once, you can find some detectable in your urine. So to get a diagnosis, this would be not necessarily following a big meal full of carbohydrates. Right, so last one is a bit of an order activity. So could I ask you to have a read through these and then pop them in the right order for me, and then I'll go through. Okay, so slightly different. We're giving you the name of the enzymes. This is off spec, so you don't need to learn it, but it is the name of the enzymes that are involved in causing the vesicles to move to the membrane. So it should look like this. 3, the osmoreceptors detect a low water content in the body fluid, so in the blood, a low water potential. That causes 5, ADH to be produced or released from the pituitary gland. Then it should be 2, the presence of the ADH is detected by receptors, so the ADH binds to the receptors in the membrane. And then number 4, this is the name of the enzyme, so the enzyme active phosphorylase is produced. That enzyme causes the vesicles containing the aquaporins to move and fuse with the membrane. And then finally, water, I wouldn't say diffuse, it moves down a water potential gradient out of the collecting duct by osmosis. It's a better way of describing. Okay, so just to show you that in a little bit more detail, so this is our aquaporins again in a bit of an animation. So there is the filtrate from the distal convoluted tubule. We're just concentrating on the collecting duct in this animation. We've got our vesicles containing the aquaporins. Along comes the ADH in the blood because it's been released by the pituitary gland in response to detecting a lower water potential in the blood. There it is. You can see in this, I like this animation that it shows a specific and complementary relationship between the protein in the cell membrane of the collecting duct cells and the ADH itself. So that activates the phosphorylase enzyme, and that causes the vesicles containing the aquaporins to move those to the membrane, causing the membranes to become more permeable. Remember, the water potential is always there because this is the medulla tissue. This medulla tissue fluid has ions that have been moved out of the loop of Henle, the ascending limb, and so around here all a really low water potential, and that means water will move by osmosis. Okay, so we're moving to the question pack now. Okay, and we've got a number of questions left to do. So you can either work your way through the questions with my next video, or you can do the questions first and then watch the video to mark the questions. Okay, right, well, I'll see you in the next video.